Hello, my name is Kathy Hernandez, and for today's presentation, we will be looking at three different chemotherapeutic drugs, methotrexate, Taxol, and Herceptin. We'll be talking about the mechanism of action and its effect on the cell cycle. Methotrexate is a anti-metabolite and a folate analog. So the mechanism of action is that methotrexate is a folate analog like aminopterin. It inhibits the enzyme dihydrofolate reductase, also known as DHFR, which is a crucial part of the synthesis of tetrahydrofolate, a cofactor involved in the production of purines and pyrimidines, which is required for DNA synthesis and cell replication. So it's important to note that the structure of folic acid is important because methotrexate and aminopterin are designed to mimic folic acid structurally, which allows them to bind to DHFR, thus inhibiting its normal function. So the target molecule in this case or a protein is dihydrofolate reductase, or DHFR, and its effect on the cell cycle is mostly in the S phase. So by blocking folate metabolism, methotrexate leads to a depletion of nucleotide pools, especially purines and thymidine, inhibiting DNA synthesis. So this arrest occurs primarily on the S phase, preventing the cell from progressing through DNA replication and induces a cell cycle arrest. So if the inhibition is prolonged, a cell undergoes apoptosis. The next drug we're talking about is Taxol, also known as paclitaxel. The mechanism of action is that Taxol is a microtubule stabilizing agent. It binds to beta tubulin, which is a subunit of microtubules, and stabilizes microtubules, which prevents their polymerization. This action disrupts the normal behavior of microtubules necessary for mitotic spindle formation. So the target molecule or protein in this case is beta tubulin, which is part of microtubules. The effect on the cell cycle is it affects the M phase. So by stabilizing microtubules, Taxol interferes with the dynamic process of mitosis, and particularly the formation and function of mitotic spindles. So this prevents the proper segregation of chromosomes during anaphase and telophase, leading to mitotic arrest at the M phase. So if these cells are unable to complete mitosis, they ultimately undergo apoptosis. The next drug we are talking about is Herceptin, which is also known as trastuzumab. Mechanism of action for Herceptin is it's a monoclonal antibody that specifically targets the human epidermal growth factor receptor 2, also known as HER2. So HER2 is a receptor tyrosine kinase that is overexpressed in some breast cancers. So binding of Herceptin to HER2 inhibits its activation and signaling. In this case, the target molecule or protein is HER2, the human epidermal growth factor receptor 2, and its effect on the cell cycle is mostly in the G1 phase. So HER2 signaling normally promotes cell proliferation through downstream pathways, but Herceptin blocks these signals, leading to a decrease in cell proliferation. Herceptin also can induce cell cycle arrest in the G1 phase by preventing the activation of cyclin-dependent kinases, or CDKs, required for their transition into the S phase. So here is a chart of the key differences of each of these drugs and comparing the mechanism of action, the target or molecule protein, and the cell cycle or part of the cell cycle it affects. First, we're looking at methotrexate, which its target molecule is the DHFR, and the mechanism of action is it inhibits folate metabolism, which disrupts DNA synthesis. This mostly affects the S phase of the cell cycle and leads to S phase arrest, where DNA replication is inhibited. Next, Taxol, its target molecule or protein is beta tubulin, which is the building blocks of microtubules, and its mechanism of action is it stabilizes microtubules, preventing mitotic spindle and function. This mostly affects the M phase of the cell cycle, which leads to an M phase arrest, which mostly disrupts the mitotic spindle disruption. So for Herceptin, the target molecule or protein is HER2 receptor, and the mechanism of action is it blocks HER2 receptor signaling and it inhibits cell proliferation. This mostly affects the G1 phase of the cell cycle, leading to a G1 phase arrest and the cell cycle progression inhibition. And these are my references. Thank you.